So now I want to turn to the real dessert for this meal, and that is the panel discussion by our three laureates. And for this year, Dr. Fraley, Dr. Montagu, Dr. Chilton. And I said, you know, who can we have that can keep them under control and yet have a lively discussion? And sitting there thinking, and I said, the only one who can do it is our council member, Margaret Catley-Carlson. So, Maggie, the herding of the laureates is over to you.
Where's Mary-Dell? She's okay. I'm going to take that one, if you don't mind. Okay. Yes, it's super. Okay, and we'll put Mary-Dell in the middle.
My feet don't touch the floor.
Something's happening here.
Is this okay?
Yes, this is fine.
My feet don't touch the floor in that chair.
Oh, your feet don't touch the floor. That's important. Okay. Can each of you just say hello, so we'll do a mic test?
Hello. Good afternoon, everybody.
Hello.
Hello, hello. Okay, so we're all good? Okay. Well, welcome. And this is going to be a really nice event because you've got these three fabulous people that we've been celebrating all week. In some set, we've celebrated also the amazing Ken Quinn and his magic staff that put all of this together. So this is a time to sit back and to have some reflections on what's really important about the work that you've done and the way we've looked at these issues this week. So we can go on all afternoon, but we lose our audience at 2:30. So that shouldn't stop us. You know, we can go on a very long time as long as we don't mind not having an audience. So anyway, Marc Van Montagu, who I've known for many years, Mary-Dell, if I may, Chilton, and Rob Fraley. Rob, he's got two B's in the Fraley, he says. So,
No, in the Rob.
That's right.
In the Rob.
In the Rob. Yes, exactly. Okay. Well, as I say, we've had a marvelous lead-in to this meeting in the panels this morning, and I think that what those panels covered gave us the ability here not to talk about a number of subjects, what could have, should have, might have been, whether current priorities meet ethical imperatives, how to meet the campaign of misunderstandings, half-truth, some truth, no truth, regulation by superstition. All of this kind of leads to gloom, even though it gets punctuated by little rays of sunshine, such as we had this morning from the Philippine announcement. But I thought that it would be a good thing as we move on to celebrate the amazing science and the amazing scientists that we're honoring, rather than going into these issues. Even if we get into them, we do. But I wanted to start by really finding out a little bit more about some of the science that we're talking about here. And I want to start with the idea that great science is a springboard for a lot of things other than the products that we have spent a good deal talking about. And we have here three great scientists, whether you're looking at the T cells, whether you're looking at the idea that bacterium can be used to move one characteristic into another plant, whether you're looking at the idea that once you can take one characteristic, you might even transfer that to something as astonishing as pesticide resistance. So, it's more than the trail that leads to a product. It's opening a whole door on a new place that wasn't there before. So I'd like to hear from each one of you. Tell me about those and the science and the people that have used your science that have moved from the springboard that you created to go in the most interesting places and the places that you're most proud of. Have you got a couple of examples? Mary-Dell, you look like you might have one that you could share with us.
My interest has been in the beginning the science of gene transfer did I do that?
No, I think you're okay.
We began this as a study in pure science. We were interested in how a bacterium could cause a tumor on a gall on a plant. And as we began our studies, there was some literature on it that seemed to show that that might be caused by gene transfer, but there wasn't any direct evidence. And our team at the University of Washington in Seattle was able to show that indeed there is a tiny piece of a part of bacterial DNA that went from the pathogen into those plant cells.
Now, what's taken off? I know that what you've done personally with your team has affected the lives of millions and millions of farmers, particularly in the corn area. What else took off from that science? There must have been all sorts of interesting things that took off from that bacterium development.
Well, all of these people are, of course, or most of them probably, are on the agriculture side and interested in the gene, in the products, the plants that have been produced. But I would like to recall one other thing that has come as a consequence of gene transfer to plants. We can use these tools to understand how plants work and understand the
I wish I could guide you on what's doing that. Stop breathing, maybe?
when I turn blue, you can
Yes,
turn
when you turn blue, yes.
But anyway, the tools have been also extremely valuable in basic science in understanding how plants work and in helping us to identify the plant genes that will give tolerance to drought and tolerance. Sorry.
Okay, we'll turn you off. That's right. They'll turn you off at source, and you can use that one.
Sorry about that.
Go ahead.
Anyway, so yeah, it's a tool not only for making products, but for making new science, which in turn will likely lead to new kinds of
So the
products.
whole idea of marker-assisted selection, where you can go in and you can look and you determine what gene is going to do what, this also came out of the very much out of those initial explorations.
Not exactly, but when by marker assistance you find a gene, we are able now to pick that gene up and change it, put it back, and prove what the function of the gene is.
Us non-scientists try and imagine a pair of tweezers that are small enough to reach in and do that.
Well,
And then when I learned, it's called gene expression, which doesn't help non-scientists at all. Rob, what do you particularly, who used your science as a springboard to go in interesting places?
Well, let me reflect on a couple of things that I think it happened so fast that we take for granted. But I think really one of the really exciting things that happened is after a lot of these basic tools were developed in the early '80s, it really just drove a phenomenal amount of reinvestment in scientific and human capital across the plant sciences across the world. I mean, you can look at the publications, and Marc used that in a slide. But I can remember back when I was looking for my job in 1980 and trying to find locations where you could do plant molecular biology research. You had to look pretty hard across universities and companies. And for me, what's so exciting is you can go anywhere now. I mean, the phenomenal institutes in Beijing. We were just talking about the scientific capital that's now being developed across Africa. I mean, for me, what is so exciting is we are seeing that next generation of knowledge explode. And I think it's in part been to the tools, the excitement, the ability to both invest, and that's cool. The other one that I always like to emphasize, and I start a lot of talks like this, because it's one of the folks who helped really pioneer, transgenics and GMOs in this tool. I always tell folks, for me, really the most remarkable transformation in science that's going on is plant breeding. I mean, these tools have fundamentally changed how we breed crops. I mean, once, the complete genomes were sequenced and mapped, and breeders had that ability to understand exactly the combinations that were being created, it has changed how you breed. I mean, you know, we can now, a breeder can find a new disease trait in Brazil, mapped and tagged, make your crosses, do your markers, and within, you know, a year, you've got a Brazilian gene in U.S. germplasm. If you tried to do that 10 years ago, it would have been virtually impossible. And that's what's changing the game. And then that leads then to a whole new wave of gene identification, the ability to pyramid genes, which is going to be so important for, you know, for resistance management. And I think particularly when we think about being prepared for the future, you know, climate change and rapidly evolving dynamics in pest populations, these are going to provide really important tools. And I always use the expression that, you know, today every breeder has the potential to be a molecular breeder because they can literally breed gene by gene. And I really think when you kind of reel this all the way back and you look forward, the investment in the sequencing, the markers, the information technology, it will change breeding. I mean, we've done a couple of estimates that, you know, the ability to precisely pick that seed off of an ear of corn, you know, the right brother and the right sister. You know, we have good kids and bad kids. You know how that works. But to pick the right one precisely, and that you want, that has that combination, can improve, you know, the efficiency of a breeding cycle by thousands of fold. And
so your answer to the springboard is more money, more interest in agriculture, which is a really good thing, and really opening the door on a totally new way.
And a real plea that molecular breeding, breeding is going through a renaissance, and the ability to apply that technology not only to the large acre crops, but to smaller crops and orphan crops is enormous. And it's happening at the same time that the cost per data point makes this applicable to virtually every crop in the world.
Astonishingly low.
And we don't want to give that one up. I mean,
Rob, I'm going to have to get over to Marc. He's going to have to add an answer too. Marc, what springboard action are you really happy about that was a result of your work, which got to amazing places? Not only, I like the pulping of poplars. I thought that was
well, that's in the application, but still for us, the fundamental science was so fantastic. When we started, nothing was known on the molecular genetics of plants. We had not the slightest information. Now we are, as Rob said, yes, that we have so many sequences and that this aspect can be done. But in fundamental science, there is still so much to do. And we hope that there is good funding for that, because now we realize you may know the genes, but the way they are expressed, the opening of the chromatin, what we start calling now more and more epigenetics, is still to be found. All the role of these small RNAs, and it's the same technology of making transgenics in the laboratory on a model system like Arabidopsis, we will bring you this information. But we badly need it. We cannot without. Everybody knows that identical twins, if it was the same DNA, the same gene, the same order, that during development, so many characteristics change. Not physically a bit less, but susceptibility to disease and character, way of behaving. So what's the difference in some types of tissues that came? Can it be reset? Because we will learn it in plants. It's not only how it works, but mostly can we reset? If a plant that is germinating has a certain stress, but some are overcoming, others are not overcoming. They were sometimes identical genetically, but it is parts that have been switched on. Can we reset them so that they now can don't remember the stress they went through and they can go? That is crucial for trees, because you cannot, if you have to make crosses that every 30, 40 years. And so
that's fascinating.
That's the type of
idea that you have to try and
of science I'm waiting for.
It sounds more like science fiction, erasing the memory of a tree about the stress it's been through. That sounds absolutely fascinating.
When we started, doing the sequence was also science fiction. My colleague Walter Fierce, when he started sequencing RNA chemically, he did it 4 nucleotides. I have 5. Now I can separate a piece of 10 nucleotides, and I do. But we went on, and that's what makes then the possibilities of that we know today on sequencing.
It's very interesting. All three of you have basically said, okay, the event was important, but you immediately went to an enormously broad canvas because, of course, that has been exactly the implication of what you've done, is to move from a particular discovery, and very, very quickly it moved to a very broad canvas indeed. So thank you for those glimpses. Now, we've got a lot of young people in the room, and I hope that some of them are going to leave this event deciding that, hey, I want some of that excitement, and I want to feel that way about having really changed the world and removing the stress memory of trees and, you know, doing some really exciting things. And I want to know, take yourself back to the time when you really, you know, when I read the bios, when you were sitting in the labs and you were you know, working presumably, like before you discovered what you discovered, can you remember the moment or the event that made you turn a certain corner to find out what you found out? Because, you know, you've got people here who are wondering, how does that happen? How does that magic actually happen? Is it just that you do it 195 times and the 196th time something turns out differently? Is it an inspiration? Is it a moonbeam that comes in through the window? What happens to turn you from a very good scientist into the great scientist making great changes that you made? Rob, what happens?
It's real easy in my case. We had a phenomenal team that I joined when I went to Monsanto. And, you know, Rob Horsch and Steve Rogers, and you know, the chemistry and the ability to link in one place with kind of one thought process, the expertise from Rob's magic fingers with tissue culture and being able to regenerate plants from cells. Steve Rogers, a vector wizard, and you know, I worked a lot with agrobacterium, and it was that teamwork that just was phenomenal.
But tell the young people, I mean, when you were trying to find out what you found out, do you keep going in a straight line? Do you make a list of all the things you're going to try? Give them some idea of what's involved in making the kind of breakthrough that...
Well, as you all know, it's only a straight line when you look backwards. But whether you're talking about your career or your science, I am famous for, we try everything we can possibly think of. And we try to design really, really good experiments, so sometimes the most important answer is no, but you have to have no in a way that you can go forward with something else and make decisions. And I think I would recommend doing that on your careers, and I'd recommend you do that on your science.
Mm-hmm. Mary-Dell, when you finally convinced that bacterium to do what it didn't know it could do, was this a straight line, or was this magic, moonbeams?
I want to go back to the first experiment we did that showed that there was gene transfer from agrobacterium to the plant cells. We recognized that this would be an extremely unusual thing if it were correct. And so we designed the experiment in a way that if it said yes, there would be no arguing. Okay, we did all the controls. And as an example, all the glassware that we used in handling the various solutions was either brand new, or if it had been used before, it was fired. We bought a self-cleaning oven at the Sears Roebuck store, and it was all fired in the self-cleaning oven. The reason is we wanted to be sure that when we found the DNA in the plant cell, if we did, we wanted to be sure that it wasn't contamination. Mm-hmm. So when I was at home in my kitchen after the boys were put to bed, calculating the results of our experiment, and saw that we had a wiggle in the curve that showed that DNA had been transferred to the plant cells, I knew right then that it was right. No argument.
So was that inspiration? Did you know it was going to turn out that way? Did you hope it?
On the contrary, I expected it to be the other way. I was sure that bacteria couldn't put their genes into a plant and have them work. Good heavens.
So that's an interesting piece of advice to young people doing a career scientist.
But the lesson is we designed the experiment in a way that we would know definitively, yes or no. We could stop working on this if it said no. Or
And
we
go
could-
on to the more profitable ways
Exactly.
of looking at it.
Exactly.
So contest everything, but don't contest gravity from too high a level. Marc, what the moment that it all started to happen, what was happening to you, for you, in your minds, and your team?
Well, the history is that we just, by looking what other people did, we were convinced that it should be genetic transfer, because these tissues, you could not think how, in those days surely, how a plant could start making these odd amino acids if there was no genetic information for doing it. So we were convinced, and then I think one of the approaches is, well, there were maybe two things. First is Jeff Shell likes sports, and if he's doing sport, he likes to win. So he could give the motivation, and it was teamwork indeed. But Jeff had been in the UK with Bill Hayes for as a postdoc, because he thought systematics of bacteria that he had to do for his PhD. He didn't like that at all. He wanted to do genetics, and he went to the UK. And there he said, I learned something. In the UK, you look how you can do the experiment, and then you say, oh, but that will take a whole week of work. Maybe we go for a cup of tea and think it over, maybe. And if the experiment can be done in an afternoon, yeah, then we will do it. And that creates another atmosphere. And I think it was important.
So your lessons were, don't stay in the UK if you want to do a particular kind of science.
No, absolutely not. Absolutely not. I think it's a good approach, but think it over and discuss, and mostly look what others do, and also for the technologies, look as broad as possible what is around. It's not now today with Mr. or Mrs. Google, you can do it, but in our days it was slightly more difficult.
What would I do without Mr. Google? Yeah. Did you two know that Marc and Mary-Dell? Did you know each other?
Oh yes, yes.
At the time of that, at the time that these discoveries came out.
We were arch
While we
rivals,
were
in
working,
fact.
we were even on the
You were
phone
rivals,
telling
yeah.
the results to each other. Yeah,
So you were rivals,
Yes.
sure.
but you were also sharing
You have to
information.
win.
But we were arch rivals, and at the same time, pretty good friends.
And do arch rivals share information? Some information? A lot of information? A little information?
I'm glad I'm in between here. See, we collaborated with both of them. I mean, I don't know about you guys.
That's right, yes.
No,
Well, I
we-
remember the story on that the tDNA was one piece of DNA, that you said, oh, nonsense, and then you said, oh, you were right, my article is ready. We didn't
But you have to recall that when the article was published, it was back to back with your
Yeah,
article.
sure. Yeah, you were. Yeah, sure. You didn't. Otherwise, we would have been angry. But now you were absolutely good sport.
Well, it's...
I think though that's you know, for me one of the parts of that period that were so magical was there was good-natured competition with a lot of labs, but you know, really remarkable, you know, exchange of information. I mean, the Gordon conferences back then and the meetings we had were absolutely remarkable. And it was you know, there was a sense of you know, wanted to, you know, shoot that first goal, but you know, there was a lot of cheering of the successes, because I think what everybody felt, I know we felt this, was that if this could happen, it was going to be big, and it was going to change how we think about crops. And you know, so you know, you're always kind of hoping maybe they didn't make as much progress, but you always wanted to see where they were going, because it was such an exciting time. And
I can remember the most fun of the Gordon Conference one year was the time that we had just made a map of all the fragments of the whole TI plasmid, And these plasmids were so much bigger than the ones that people had worked with in the past that you couldn't map them. Nobody knew how to map them. And I came to the conference and told Jeff Shell, we have mapped the TI plasmid. And Jeff said, you can't do it.
That's his plasmid, yeah.
Now, actually, that wasn't the most fun, Mary-Dell. The most fun was when Mary-Dell was chairing the Gordon conference and she ordered somebody to break into the bar. But that was the most fun.
Well, I confess to a terrible thirst at times.
So the lessons are for young people, be competitive. It's really important, be competitive. But remember to shake hands afterwards and be collaborative. And if you break into the bar, don't get caught. Okay? And don't have friends with long memories that remember 30 years later who broke into the bar. I mean, this is terrible. It's a good thing the statute of limitations in this.
That's right.
Let me circle around a little bit more, and I'm coming just to the edges of acceptability issues, which I said we weren't going to talk about very much. When the Cardinal was talking yesterday, he was talking about the need for advancements and changes to be seen to be benefiting the whole gamut of players, the companies, the academy, very poor people, all the rest of it. If you ask yourself, what kind of product or what kind of product line, is there anything you can think of that would be likely to move the genetically modified plant into a greater level of acceptability because they would be appealing to would find a place in the heart of just about everybody, because it was a very good panel today talking about, what was that word? Bias? Yes, confirmation bias. And, we're really up against things that don't respond to evidence. And we've got to, if we're going to get over this, we've got to find things that change the emotional appeal of these products so that, finding something that people really feel emotionally, we should have that. We should be working towards something like that. That would be so important. And it's really asking for a big leap to asking scientists, because we're saying, the evidence is one thing, but we've got to fight this battle on more than just the evidentiary basis. What could be the kind of development that might move us in that direction?
We've already done it. We've made golden rice. We have made a strain of rice that produces the precursor to vitamin A. It could be a great boon to African children. We lose 1 million children per year to this deficiency.
That's out, and it hasn't done it. It
hasn't been accepted.
No, it hasn't done that warm and fuzzy.
It's not out. It's in the. It's
Yeah,
still,
well, it could be out in the
blocked.
Philippines. Yeah, exactly. But anyway, Marc, do you have?
Yeah.
Is it out? Where is it?
No, I say it is not out.
It's
not
out,
It's
yes.
on the field trials that are then destroyed by the activists
Yes.
who are paid to do it.
Yes, and we got that in spades this morning. Yeah.
Yeah.
Can you think of any kind of
Sure.
product area that could change the emotional or the
Sabotage.
They're pulling up my yes. Could change the way these are seen.
Well, I think in the laboratories there are hundreds of examples that are ready. We have seen it, the papayas in Hawaii, they have been in the field and still people are protesting against it. We have the potatoes that are resistant against potato disease. But all this is in laboratory, and nobody has the money with the regulations that are around to bring it to the field. It's not, and so the battle has to be at two levels. People have to realize that, okay, we know due to the confirmation bias, the way we are thinking, people will have the need for having the product. But you could already, by reasoning, say that all these things can be done. And they are really like products, just like in the old days in the 19th century when the Luddites were destroying the machines. if you know that there will be a whole economy, that the machines will really, the production system will be different, that so much will be changed. We remember in our area in Flanders, in the beginning of the 20th century, around 1910, people were living in shacks, were having 10, 12 children. At 5 they had to contribute to the income. Nobody could go to school, but industrialization has all changed that. And from 1910 in 1950, it was completely different. Nevertheless, people started destroying the machines.
No, people don't like change.
Yeah.
No question about
so
that.
this aspect has to be considered. So we have already all the arguments in the lab, and Greenpeace knows that they just have to see that it cannot reach the public. So it's not the science that is the block. The science has already.
No, that's what I started off by saying.
And the products could go, but nobody gives them the authorization to go. You have the regulatory, you have the financial aspect, all the controls and the super controls and the recontrols that one has to do. So that's where the block is. But indeed, psychologically, people need the products. So if we have to insist,
they don't think they need it. That's the
no, they don't think. But once they use them, they will see, and we have to try to find then the best example that there cannot without, because till now agriculture is so essential that people try to, and the stresses have always been there, so we learn to work with the most essential things. But if we see a plantation forest, because plantation forests are all made from identical clones in cell culture, nothing to see with GMOs, these clones. But these forests are there. They are there in Oregon, and they are there in the Seattle area. And there are some beetles that coming in a record time can cross it out. Then people can understand. We should have other solutions.
Yes.
But okay, these trees will then also not be immediately.
I apologize for these beetles. They came down from Canada. So, yes. Rob, can you think of a product or of something that could appeal to the emotional intelligence of people and try and get around some of these barriers? Because the panel this morning was very clear that evidence is not the answer at the moment.
You know, we obviously haven't found that product yet.
Yeah.
And we've had some pretty good ones that have increased yields, and we've had some now that are protecting crops from drought. What we haven't had is the product that has unambiguous benefit to the consumer without going through the dynamics of food security or production or yield or benefits that are generally targeted at the farmers. My guess is when you get into these consumer traits, and we've worked a lot on looking at vitamin enrichment and colors and pigments, I think a lot of that's going to come again from the molecular breeding area. And one of the areas that I think we're going to see just a phenomenal set of advances in a few years is, most now of the major vegetable crop species have been sequenced and mapped, and all the genes for the nutrients and the flavors and the consumer appeal have all been mapped and tagged. And what we'll see, I think, is just a phenomenal proliferation of fresh fruits and vegetables that have different appeal. They may not be GMO, but they're going to be some of the most remarkable products of genetic modification. I always like to use the example of when you walk into a grocery store today, and the first thing you see is, first of all, generally the produce area is right in front of the store. And when you walk in, I mean, the colors, the availability is remarkable. And that is all just phenomenal advances in genetic modification. Yes. And that seems to, I think, in my mind, be the big winner. It's hard to focus on something else. I mean, we've looked at amino acids and lysine. We've looked at putting omega-3 fatty acids into soybean. But there's always alternatives. There's different approaches. I haven't found the magic bullet yet.
Mm-hmm. No, I don't imagine so. But anyway.
Yeah, I think
you want to add a final word on that?
The initial products that we made were ones that you could create with transfer of a single gene. Resistance to an herbicide, one gene. okay? A BT that makes it resistant to an insect, one gene. So the technology in the beginning enabled us to transfer 1 or 2 genes. We can transfer many now. We can transfer 10,
Yes.
15, 20, and the number will rise. So I think we will see entire pathways that synthesize chemicals, new
Wow.
useful chemicals, pathways, whatever.
Well, you're very much leading into my next question. But before I leave this one, I'm going to give you the suggestion. I'd like a 6-cheese lasagna with no calories. Okay?
Now you know how much recombinant chymosin would be in there. I think one of the best chances, for that kind of public awareness is going to be in the response to drought and some of the climate change. It may not be a direct consumer benefit, but everybody in the world understands drought.
Yeah.
And I think that has a lot of appeal to people.
Some of the things that MS Swaminathan is doing, with really some of the poorest people in India that are being menaced by salt intrusion, and developing plants that will be able to stand up to that saline intrusion and even increase yields and increase the prosperity. I think that's the kind of thing that will grab hearts and minds in a way that other products don't yet. But you've started, and the next question that I was going to go, so I'm going to give the microphone back to you. Where do we go beyond the GM techniques that we are today? And you've both already started into that. So can you start painting your canvas about where we go from here and today's technology? What does the future technology in plant science look like?
Well, what I find interesting is that agrobacterium did more for us than just put genes into plant cells. It inspired us all across the science that if you could get a gene in there somehow, it would be able to get itself into the chromosome of the plant, and it could express to create the new trait. So people have developed different means of adding genetic information to plant cells, not just by agrobacterium. I stand by agrobacterium, it's still the best way. But people coat DNA genes onto little bullets made of gold and fire them, shoot them with a gun into the plant cells. And that works. It gives you genes that are scattered across the genome of the plant in more complicated ways. Agrobacterium puts them in one place much more tidily, usually. Anyway, there are various ways to put genetic information into the plant cell, and I suppose some of those may be exploited in the future. And I think that strings of genes that have a whole pathway will be put in there. I think multiple single gene traits will be put in.
Give us a for instance.
Well, we need to have different active principles to combat the same insect so that it doesn't just mutate in one jump to be resistant to our plant. So that's one reason we need multiple traits. But then also we need an herbicide resistance. We need resistance to fungus. We need resistance to all kinds of things in the same plant cell.
So you would just develop a whole string of resistances which could be put in by your...
Yeah, we call that stacking the traits.
Stacking the crates, yes.
Trait,
Okay.
trait, not
Traits,
crate.
crates, yes, okay.
Yes, whatever they are.
Really high.
Yeah.
Yes.
Yeah. And you can make a plant with multiple traits either by making a plant with one trait and another one with another, and crossing those together and finding the two of them. And you can do that with yet a third trait and a fourth trait. But when you do that, the traits are all over the genome, and so the breeder has a problem breeding with those. They want to separate into the progeny. So perhaps a better way is to stack the different genes into one construct, into one DNA molecule, and put that in. So we've been asking agrobacterium whether it could put long stretches of DNA into the plant cells. And happily it says yes. It says, I don't do it nearly as efficiently, but I do it.
I do it.
I do it.
See, you should never have doubted it 30 years
You're
ago.
right. Absolutely.
Marc, what's the future in post, sort of beyond GM plant science or the next?
Well, with all the technology that we have worked out to study the plants, now we can do metagenomics from the microorganisms. And all the plants grow the roots, the leaves. It's full with bacteria that never have been studied. Now we can do delivery of products through this bacteria. We can study which one is the best to do that. We can use the metagenomics for making new soil much more rapidly. Arid areas, the problem is the soil. So the molecular biologist will in the future work much more closer with the soil scientist. Soil is not inorganic chemistry. Soil is a living organism. So many organisms there are there that never were studied. With all the technologies that we have used for the plant, we will start doing that. And then people will see also rapidly progress in agriculture if we can adapt the soils.
Rob, the future?
I think one of the areas that, it is tantalizingly close, but we can't quite declare victory yet, is in this area of being able to use RNAi molecules to modulate plant activity. So that's a whole level of science. Back when Mary-Dell and I were learning biochemistry at the University of Illinois, the gene made the RNA, which was the intermediate that made the protein. But, a few Nobel Prizes ago, people discovered that RNA itself was regulated by small little molecules called RNAi. What we've found out is that you can actually manufacture these RNAis. They might only be 40 or 50 nucleotides long, and you can deliver them to plants and elicit very specific reduction in expression of a target gene. And the beauty of it is it gives you a lot of the specificity of biotechnology without a transgenic organism. And a couple of examples that we've worked on, if we synthesize these small RNAis to a plant virus, and you have a plant that actually even is undergoing virus infection, if you spray these RNAis on the plant, it will cure the plant of the disease. And we've also been able to show that for a couple other areas. And one that's close to my heart is, in the 40 years that, Roundup has been used as a herbicide, there's 3 or 4 weed species that have become resistant to it. And what we've been able to show is we can sequence now the weed and understand the mechanism biochemically by which it created the resistance, and we can design an RNAi molecule that knocks out the resistance mechanism, and now the activity of the herbicide is restored. And that's pretty cool. But the area that is really special is in, I think, this area. And it's really cool, and the science is still preliminary yet, but everybody is very concerned about bee populations.
Yes.
And colony collapse disorder. Yeah. And there's a lot of theories, but most of the scientific consensus points to the fact that the bee gets a mite infection, and the mite transmits several viruses into the bee. And we've seen that you can now design these RNAis that can knock out the mite and start to knock out the replication of the viruses. It's still complex because there's a lot of different virus strains, but this is pretty cool when you can use the power of very precise targeted gene expression with a naturally occurring molecule that can do some pretty neat things.
I think that's more important than a 6 cheese lasagna, no calories even. Are you saying, though, that what I hear you saying is that some of the things that are now being done through genetic gene transfer from one plant or one entity to another plant might be done in the future by another kind of science, another kind of technology, another kind of technique?
Yeah, I think they build on that same knowledge, but they could be done with different approaches. I mean, the way I look at it, if there's any way I can solve a problem without spending $150 million of regulatory costs to develop a GMO product, I will try breeding, molecular breeding, anything else I can do. But there are just some things that GMO technology will do better. But there are these other areas where I think there are remarkable opportunities. I mentioned the breeding before, but the RNAi approach could be very useful for disease and insect and very specific regulation of genes that you may want to reduce for ripening. So one of the clear applications is relooking at the ripening response or the over-ripening response, and using these RNAis to specifically knock out that ripening mechanism, at the crucial time.
Exteriorly.
Exteriorly. So
Interesting.
the way it works, just so these are very small molecules, and you can formulate them that they will actually penetrate through the leaf, and there will be enough of that molecule to elicit the biological activity. But it's not a permanent gene transfer. It's a temporary effect. And it's really cool.
So that could be an interesting backdoor for some of the problems that we have been talking about.
From both a cost and a market access.
We're going to be losing our audience to the soybean farm soon enough. So I want to give each of you, though, before we close this part of your amazing 4 days as being laureates, and the chances that we've all had to get to know you better, and the opportunities we've had to explore 3 of the very interesting minds of the 21st century. So leave you a couple of minutes to say what you'd like to say as we close this session of the World Food Prize dialogue and the Borlaug dialogue.
I'd like to just start, first of all, by thanking you, Ken, and your team. I've had the privilege of being part of a lot of cool events, but I don't think I have ever seen anything that's been so not only extraordinarily well organized, but the detail and the nuance and the connectivity is really remarkable. I mean, it's really special, and I appreciate that. And I'd like to thank everybody here. This is a remarkable. Thank you. This is a remarkable experience.
Collaboration.
I told you I collaborated with both these guys. So. And I just want to say thank you. Somebody has to put the first shot in the goal. I mean, but really appreciate it. I've thoroughly enjoyed it, and my sense is it's been a pivotal conversation in terms of shifting and rethinking views. And the thing I would ask is, the science is so important. It is so applicable to so many of the problems, and the barriers aren't the science. The barriers are the policies and the politics, and that is really, really worth rethinking. And I'm not going to debunk all those myths that have already been debunked. I thought the panels did a great job, but we don't yet appreciate the power that this technology has for the future, and to think that we're either regulating, over-regulating, or in some cases blocking the adoption of tools that are going to be utterly transformational. It is. No, I'm going to keep pushing.
Well,
it's too exciting.
We've got to work on all the barriers, including the emotional one, not necessarily for the scientists to tackle that one. What is the emotional appeal that we could use that we're not using with sufficient clarity and force now? Because if the difficulties are emotional and non-rational, then we've got to put some of those in our armamentarium.
Yep.
Mm-hmm. Absolutely. A last word, Mary-Dell, or last several words?
Well, I certainly echo the things that Rob has already articulated. We certainly thank you and Ken and the other organizers of the whole operation. It's been a joy to be here this week. I have never seen so many supporters of the technology in one place. I think it's a wonderful technology, and I hope in my lifetime to see it not only accepted but embraced throughout the world. We need it. We really need it. And I think we have our work cut out for us. Some of us, and I point to the gentleman on my right especially, are good at talking with people and persuading. We need to talk to some of the leaders of the organizations that seem to think they're opposed to us. For example, the Sierra Club. I'm shocked that the Sierra Club thinks that we're not green. We are. And someone needs to talk to them and get it across. I don't know what the problem is there, but there are more organizations that ought to be on board with us that just aren't. We need to talk to our school teachers, find out what they're teaching their kids in school. we need to do whatever we can to spread the word. And I'm speaking, of course, of what we can do in this country, but I think Marc will address what we need to do in Europe and worldwide also. And I want to just thank the audience for your wonderful enthusiasm, and thank you for your wonderful questions. Okay.
Marc, the one who gets to not just battle from the trenches, but living in the trenches as you battle.
That's not a comment on Belgium.
First, an important point that Rob was saying, and that maybe was not really taken up directly. That is, if a major company like Monsanto, for each trade, they have to spend $150 million. They are not the machines of the earth. Why should major companies, they have to survive and have made their products? So we know that we have to organize that the public sector does it. But meanwhile, since they have to look for alternatives, what they are doing is fantastic, and it's developing with brute force approaches that we then in the public sector could not do. But the whole RNA technology, because it's at the basis also in epigenetics and in the gene silencing that was discovered in the 1990s period, so that it again comes all together. So that what the public sector does and what the private sector then brings in as new technologies is fantastic, and that will make us progress in agriculture. But there is no way out. The DNA technology has to be used because the problems will not be sufficient only because breeding is slow and a lot of the techniques is slow and still have to be applied, and there can be problems coming up. So we urgently have to see how can we communicate to the society, saying that it is a bias on beliefs, that what the rationality is, and again, since the people don't listen always to the rationality, you have to give the information at the same time the products. So it's complex, and we'll see what we can do.
Okay, how many of you? Yes, indeed. How many of the young people in the room, the people who are just starting off in their careers, have got their interests raised by this week's discussions on biogenetics? And way up, come on, let's. Hey, fantastic. Okay, great. Well, I hope that the lesson that you might have got from it is that part of the battle has to be in the laboratory, but part is also in media, exactly as has just been said, in teaching, in understanding what makes people form ideas, in looking at these. So if your answer to my question was, well, I'm not that good at science, I couldn't get in there, there's a lot to be done that isn't going to be done at a laboratory bench or at a test tube. So therefore, if you find the arguments compelling, and if you don't, I don't know what you're doing here for lunch, but if you find the arguments compelling, remember, there's a role here for an awful lot of players, and you've got to shape the world, because if you want these advantages that have been set out very eloquently as a very new kind of world, if you want these advantages in your world, you're going to have to battle for them. And the battle has to be fought on a lot of fronts. So what a good thing that the initial warriors are as wonderful as they are, and that they've been with us today. So thank you very much, and back to Ken.
Thank you, Maggie. Thank you. So if I could ask our laureates to stay, and you to go. That's yes. So I'm for those of you who have watched power transfer, you're and part of governance and everything is who has power and who doesn't. And whatever I've asked the laureates to do all week, they always do it. My power is about to end because we're going to have the ceremony where they're presented their diplomas and, more importantly, their checks. And that so.
There's only chairs on that side.
Yeah, that's right. So, John Ruan, could you come up, John? We'll do this. And what I'd like to do is, if I could ask the three of you to stand, and Nora's going to push the chairs back, and maybe you could come over here with me.
All right.
To start. Mary-Dell, you stay right there. All week you've been a group of three,
Yep.
so we wanted to do this individually, and and you might want take your badges off. You don't need them to get in and out anymore, and yes. And the students are all. You're not being boycotted or anything. The students are going off to for their farm visit, and then the soybean folks. But John, why don't you go to the other side?
Oh, okay.
And here you can take the checks with you, and that. So Dr. Chilton. Huh? Well, you signed them. And if Nora's got them for Dr. Chilton, and that. So we'll come out, and John will. Yeah. Yeah. Got the right name on it, does it? Yes. Yeah. So Julie and Jeannie, Norm Borlaug, one of the first things he asked me to do, he said, there needs to be a diploma. You get a diploma when you get the Nobel Prize. And we just had the sculpture that Al Clossie had created. So we created this diploma to go with it. And I had to make one up for all the past laureates who hadn't gotten one. So congratulations, Mary-Dell, to you. Yes. That's right. So if you want to come over here, and then we'll let Rob go. That. And
It's pretty heavy.
yeah. Mm-hmm. Yes. I was hoping you weren't going to look. Michelle, we need to raise more money so we can.
We better talk to John, but in all seriousness, I just wanted to make sure everybody knew that one of the things I talked about a few days ago was in my appreciation of the World Food Prize in honor of Dr. Borlaug, I'm contributing my funds and some matches to University of Illinois. We're going to create the Fraley Borlaug Fellowship to train young women in molecular breeding.
Well, I guess in that case we'll put something in the envelope. Okay, and that. So, yeah. So if you want to come, Rob, come over here. Don't go away. We'll go back and have another threesome picture. But Marc von Montagu. Yeah, that no. There's one in there for you. So now why don't we have the three of you go out and each hold your diploma, if that's all right. We can have a group picture and. Is there room? We'll pull the table out of the way here. Yeah, it's going to line the frames up now. This is just like a gene sequencing, You got stacking the diplomas, something like. You got to make it sound like you know something, even though you don't. Yeah. All right. Another round of applause for our laureates.